All attendees are in listen-only mode.
Hi, and welcome to Advaita Bioinformatics webinar presentation. My name is Andrew Olson, and I'm the Vice President for Business Development here at Advaita. As a matter of housekeeping, just to let you know, everyone is on mute, but you can ask questions using the question box in the control panel for the webinar. And if some of you could just acknowledge that you can see us or see the screen as well as hear us, I'd really appreciate somebody just saying, yeah, I can hear you, or something like that in the control panel. That'd be great. And then obviously, as you ask questions throughout the webinar, please feel free to use that control panel to ask those questions, and we will be answering them at the end. So, yep, it looks like people can hear us, so that's great. Thank you very much. Today's speaker is Dr. Cordelia Ziraldo. She will be reviewing what's new in Advaita's latest application, iVariantGuide. iVariantGuide is a powerful web-based application designed to help you quickly identify and visualize variants and understand how they may be affecting various systems. Dr. Ziraldo is the product manager for iVariantGuide and is also a computational biologist with expertise in systems biology approaches to understanding inflammation and immunology. Her graduate and postdoctoral research was focused in two areas: building mathematical models of biological systems to generate testable hypotheses, and two, generating biological insights from clinical samples using statistical and machine learning approaches. Dr. Ziraldo holds degrees from the University of Chicago and the University of Pittsburgh. At Advaita, she is leading the launch and development of the iVariantGuide application and is excited to share the powerful computational approaches with a wider community of biologists and clinicians. So with that, I'd like to turn things over to Cordelia, and she'll take you through today's presentation. Cordelia?
Thanks, Andrew. Hi, everybody. Welcome. Thanks for joining us. Today, I am really excited to show you what our developers have been working on really hard this summer, and show you how their hard work has really paid off in terms of what you can get from iVariantGuide. So, briefly, we'll look at what are the new features of iVariantGuide and how can you find significance in your variant data using iVariantGuide. And then I will demonstrate that with a real data set and save time for questions at the end.
And just as a matter of housekeeping, this is Andrew again. We are recording today's webinar, so if by chance you have to leave us early, we will be sending you a link with the recording of it afterwards.
All right. So if there's anything I want you to remember at the end of today's presentation, it's this. iVariantGuide is an all-in-one platform. We take you from sample variants all the way to biological insights in a single place. And even though it's so comprehensive, it's easy to use. Before you know it, you're quickly immersed in your data. It's quick to get started, and it's easy to navigate. It's a powerful tool built on rigorous statistics so that you can be confident that the answers you're seeing are meaningful. And our web-based platform is convenient. You can do all of this from your couch or favorite coffee shop just as easily as you can do it from your office. So there are a few core functions of iVariantGuide, several of which have been updated, and I'll go through them in more detail as we continue. But I just want to lay them out for you here so you have a sense of the outline. We have comprehensive annotations and interactive graphical filters, which you'll see, which can be saved as presets, which are really powerful functionality that allows you to do pathway analysis and gene ontology analysis to look at the biological context of your variants. So what do all those features mean? It means that you can quickly get straight into your data and start analyzing it. You can get deeper insights. You can feel confident of the answers that you receive, and then you can share it with collaborators, share it with other people in your lab. Let them see what you've found very simply, and then get right back to the bench very, very quickly. So here is an example of what a workflow looks like in iVariantGuide. You start by uploading samples, which takes about 1 minute. And once they're up, you can double check that you're getting what you expect by looking at summary statistics. From that, you can create a report. So a report is based on a sample you uploaded, and you also give a title and description. This is where we annotate the variants. It takes about 15 to 20 minutes, depending on how many variants are in your file, and you'll get an email when it's done. And from that email, you can go directly into your report and start exploring. You can use our interactive filters to select variants of interest and then save those filters set as a preset. And from there, you can also look at variant details, including pathways and gene ontology terms and diseases that are specifically associated to that variant, as well as the predicted impact of that variant. Then you can we also have the pathway analysis and the gene ontology analysis that allows you to visualize your variants in a biological context and start to generate a systems biology view of your data, more hypotheses about what's really going on. And finally, sharing. We allow you to share all of your reports electronically, as well as all of the figures are downloadable as publication-ready PNGs or SVGs. It's up to you. Okay, so these stars represent the areas of the application that have been updated. As you can see, our developers were very busy. So just to review, the annotations that we provide are mostly from public databases. But what Advaita does is it brings all of these different levels of information together into a really convenient package that allows you to really leverage all of those databases to their maximal capacity so that you can really see what's going on in your data. And then based on those annotations, you can select variants of interest using our graphical filters while screening against variants that you might not necessarily be interested in, false positives or noise or less impactful variants. So here's an example of a filter cascade where we start with 34,000 variants, and by applying selections across 3 filters, we quickly get down to 293 variants of interest. We could save that set of filters as a preset, and in fact I called it novel deleterious mutations. I could take that same preset, novel deleterious mutations, and apply it to a new sample. So this other sample has 110,000 variants. By applying exactly the same settings, we filter down to almost the same number of variants, actually. 371 for this one. So once we have our variants of interest, we can identify significant pathways and visualize those variants on the pathways via the impacted genes, as well as gene ontology terms. So we allow you to visualize impacted ontology terms and find the ontology terms that are impacted using the pruning factors, which I'll show you. So one thing I want to say is, how do we really get from variants to significant biological context, significant pathways, and significant go terms? What does it mean to be significant? So we start with a ton of variants from our sample on the left-hand side, and somehow magic happens. We get to significant pathways and go terms on the right-hand side. The way that works is you apply filters as a preset, and that is what defines your variants of interest. So you apply your preset, you have your variants of interest, we still don't have pathways. What do? That's when we bring in the domain knowledge. This is the systems biology perspective. We use known pathway interactions and gene ontologies to look for how impacted genes are associated with known biology. There's so much knowledge out there, of course we should be leveraging it. And from there, we can do some statistical calculations and find what's truly significant in your data. This workflow, this process, can be applied to the same sample many times with different presets, or to multiple samples using the same presets, and you could end up with the same or different significant pathways depending on your selections and your starting material. And I'll demonstrate how that can change when we look at the live demo. But the takeaway really is that presets are central to every analysis in iVariantGuide. You must define your variants of interest before you can calculate significant pathways. And a preset is sort of like a lens or a prism that allows you to bring variants of interest into focus and look at your variants from multiple perspectives. Presets also, in addition to that, they have some bonus convenience factors. They make your analyses reproducible because you don't have to remember all the filters you applied when you come back. You can simply apply the preset. Extensible because you can apply the same preset to multiple samples. And time efficient because you don't have to go through the process of selecting every single choice over and over again. You just one click and the preset is applied. And now with our improvements, you can share reports with a preset already applied, meaning that when your collaborator opens up the report, the variants are already filtered there and the results are right in front of them. And you can also set a default preset for each report so that you don't have to remember which one you associated with which sample. Okay, so let's try it out. The data is from GEO. This is mRNA from breast tumor tissue. It was sequenced using an Illumina HiSeq. And we're gonna try to see which genes are impacted by the variants and what pathways and functions are predicted to be impaired in this data. Okay. Here we go. So we log in. And all I did here, when you log into your account the very first time, there is a demo data set waiting for you to accept it. So what we did from Advaita is we shared a demo data set with everyone, just the same way you would share a data set with a collaborator. This is what a collaborator would see if you shared with them. It's exactly the same functionality. You can accept that share, and then the report shows up in your list of reports. And that's what this first one is. It's the demo that was shared with this new account. So I click the link to go in, and as you can see, the first thing that happens is we get some onboarding. These are some slides to orient you to some of the important features in iVariantGuide and show you how to use them. Once you've gotten oriented and you're done with this, you can just click Got It. If you never want to see it again, you can click this box, Do Not Show This Again. And now you can see, here we are in this report. This is our variant table with information from the VCF file and from the annotations that were applied. So you can look more deeply at any one of these records just by clicking on it. And this is the variant details for this specific variant. And it's going to show you what are the effects to the transcript and what are the predicted impacts. There's one possible missense variant here, as well as it will show you diseases that are associated to this variant, and you can click this menu to get links out to the ClinVar record, as well as more information from a variety of sources including MedGen, OMEM, SNOMED, and ORPHANET. Some variants, depending on what disease is associated, will also have links to gene reviews. We also see the pathways and gene ontology terms that are associated to the genes impacted by that variant. So also you might notice that when we opened up this report, there was a preset already applied. Up at the very top in this gray bar, you see the total number of variants in this file was 110,000. And with this filter, this preset called clinical evidence applied, we are down to 66 variants. So let's just look and see what those variants look like. We can open up our graphical filters and it'll give us an overview of this data. And you can see we have all SNPs. We did not select SNPs in the preset, but based on our other selections, there are only SNPs left in our data set. There are mostly homozygous variants and a few heterozygous variants. And most of these are occurring in exons, which makes sense given that this is RNA-seq data. If we wanted to filter further, we can do that simply by clicking on the sections of the graph that we're interested in keeping. We can also click the title for a drop-down menu that allows us to make multiple selections at a time. This data set is pretty big, so it's taking a minute. All right. So as you can see, we are getting warning messages because we've changed the filters and we have a preset applied. So we could just save a new preset with these settings by clicking Save Filter As. And this dialog pops up, which will remind you all of the selections you've made. You can make sure they are what you intend to keep, and then give it a name and click Save. Instead of that, I'm just going to select a preset that I've already generated. So here's one called RNA-seq deleterious. I can double check that these are the selections I want by clicking this info button. And it tells me I chose variants with impact high, moderate, or low. I set a minimum depth and quality threshold just to filter out false positives, and these happen to be the same thresholds that were used in the paper. I also am looking for homozygous variants because I want those that are really going to have an impact on the function, and we are looking at exons only given that it's RNA. Okay, so So I just looked at info, but I did not actually apply the preset. I have to click the name of the preset to apply it. And now you can see that we're up from 110,000. That filter, that preset gets us down to 2,600 variants. Let's go ahead and look at what types of pathways are impacted by this set of variants of interest. So now when I click the pathways button, it's going to generate a list of pathways, and we're going to apply a correction factor. You can choose how stringent you want to be here. Bonferroni is the most stringent. False discovery rate is slightly less stringent, but still limits the probability of a false discovery. And now you can see the pathways that are left in this list of significant pathways. I think this list actually gives us quite a bit of information about our data, even if you just look at the names of the pathways. So we know that ECM receptor interaction is a lot of integrins and collagen, and similarly with actin And focal adhesion is a pathway that's really important in cancer, so we're not surprised to see that come up either. Let me show you what one of these looks like. So when you select the pathway, the details will come up on the right-hand side, and there's a description of the pathway here at the top, and then the diagram. We show you the number of impacted genes that are associated with the pathway that have variants in our set of variants of interest. And we can zoom so that you can see more of the pathway. All of the colored nodes on this pathway are colored because there are genes that are impacted by variants of interest. So for example, if I hover on collagen, you can see there are actually 8 genes here that are part of this node that are impacted by a variant in our set of variants of interest. And if I wanted to only look at the variants that were impacting collagen 1A2, for example, I could select it with checkboxes, and our variant list will update accordingly. The colors correspond to the predicted impact on the transcript. So red is a predicted high impact on the transcript, which is annotated by SNP-F. So we use SNP-F for our impact prediction. And then so red is high, orange or yellow is a moderate impact, green is low impact, and gray is modifier. It's kind of difficult to see gray, but here's one, versus white would be no associated variant with any genes on that node. Okay, so below the map, there are the table of variants that are associated with the genes on this pathway. And then below that, there are microRNAs and drugs associated with genes on this pathway as well, and references. So similarly, we can look at gene ontology terms. And again, we start by simply observing what terms are in the table. And as you can see off the bat, there's a lot of really general terms here. Organelle, cytoplasm, intracellular, that's not giving us a lot of information. So we can choose one of the ontologies to focus on, like molecular function. And when we do that, still not a ton of information. Binding, catalytic activity, we're not really sure what's actually going on. But Advaita provides correction factors. And what these will do is help you to find the most specific terms that are still significant. So both elim pruning and weight pruning are algorithms that traverse the ontology tree in order to find the most significant terms with the most specificity. So if we choose, for example, elim pruning, all of a sudden we get a better sense of what's going on. The most significant term is now poly-RNA binding, and then ATP binding, which makes sense given that we had all of those integrins in the pathways. So here is what the ancestor chart looks like for this term. And we can switch to any of these other terms by clicking on them. But you can see how this term sits in the tree. And again, the colors refer to the number of genes associated to that term and the degree of predicted impact on that gene transcript from the variant. And then again, we have our variant table below. And here we show parent and child go terms that are directly connected to the term that we're exploring. And then again, we show references. So the other thing is that we talked about how presets are like a lens into your data. So, and we can calculate Go terms or pathways based on those variants of interest, which means we could do this for multiple presets. So say I wanted to look at splice variants instead of these exonic variants and look for which Go terms had impacted genes associated to them, and see if maybe there was a completely different set of functions that were being impacted by this other set of variants. So I will go down to splice variants in the preset table and click on Go terms. And here we are. We need to, again, look at molecular functions so that we're comparing apples to apples. And we'll use elim pruning again. And what's interesting to me is that the first most significant term is still the same, even though these are mutually exclusive sets of variants. And you can see, again, here's the ancestor chart. Now the coloring is totally different because it's a totally different set of variants, which means the predicted impact on their associated genes is going to be completely different because they're different. So, and splice variants tend to be predicted to be high impact. So again, this is a new variant table associated to this same ancestor chart, but everything below that will be similar because it's the same term, so same parent and child terms. But then after the first Go term in the chart, we see that there's some slightly different things coming up. Protein kinase activity, ribosome, things like that. So once you've explored and you feel like you really have a handle on what's going on, you can set a default preset in this menu here by clicking Set as Default. And now the next time you come back into this report, this preset will be selected as the default preset, and all of the variants on the main page will be filtered according to this preset automatically. Okay, so the last thing I want to show you here is sharing, which you can't do with the demo, but you can do here. And this little icon, which is available also inside the report, allows you to either share the report by email by typing in someone's email address, and then this checkbox allows you to control whether or not they can reshare. You can also choose a preset to include or not. So if I wanted to, let's say I wanted to share it with the RNA-seq deleterious preset that we looked at the beginning. Even though it's not the default preset, we can still attach it to the report and share this preset with this recipient. Okay, you can also, let's say I choose not to do that. Instead, you could also share by a public link, and this is going to be for reports that you generate yourself. You can choose to share by a public link, so if you're ready to publish and you want people who read your manuscript to be able to see this analysis in iVariantGuide, you could share a public link in your manuscript. Okay, I do want to show you all how easy it is to upload data. So from the reports page, there's this nice, beautiful green button that says Create a New Report. And the first time you have a couple of VCFs that you could actually download to try out. So if you don't have one that's ready and you've looked at the demo but you want to try from the beginning with upload, you come to the upload page, and now there are some example data sets right here ready to go that you can download. And that way you can try out the uploading process all at once. So you start here at this green arrow, upload a new VCF, and then you simply select the assembly and browse to choose your files. And I'm not going to do that. But as soon as you've selected a file, you click upload and you're ready to go. And then from there, you can select a sample that is in your sample repository to generate a report. And that's it. It's really, really that easy. So I really encourage you guys to try it with your own samples. It's completely free to upload and explore your own data. All you need is some data and an internet connection. So thanks for listening. I'm really glad that you all are here, and we're happy to take questions now.
Great, thanks, Cordelia. So if you do have any questions, please use the question box in the control panel and submit them, and we'll go through them as best we can. So looks like we have a couple questions coming in here. So first one that we have here, is there a printable summary in iVariantGuide like you have in iPathwayGuide?
Right now, we allow you to download every single table and figure individually, but there is a printable summary report coming imminently. And so watch your email for when that's announced. But it's coming very, very, very soon.
Cool. We've got two questions that are very similar here. I'll kind of knock them both out. So what is the format required for the data to be uploaded? The other question is, do you support zipped VCF files?
So yes, we support zipped VCF files. We will take either .vcf or .vcf.gz zipped files.
Okay, cool. Let's see, I have one. Can we look at human, endogenous retroviral RNA expression?
So this tool does not allow you to examine expression levels. We're simply looking at the sequence of RNA, if I understand the question.
So by the way, if that did not answer your question, feel free to expand on it in the question box. We may not get to it in this session, but we will follow up with you afterwards, okay? Next question is, can I use metabolite concentration data sets instead of using gene-based data? Example is mRNA sequencing data, question mark.
So again, we're looking at variants, which are mutations in a sequence. And so this application is specifically for analyzing the types of mutations you would find. And you can find those in RNA or DNA.
Okay. Here's one. This is a rather general question. He says, what is the major differences between iPathwayGuide and iVariantGuide?
So iPathwayGuide, as many of you know, is our flagship product, and it is designed for the analysis of transcriptomic and proteomic data.
Expression data, right?
Basically,
yeah.
Gene expression data, and looking specifically at differential expression. So how do the genes or gene products that are present in one sample different from, or one set of samples different from another set of samples, and what pathways could be involved in leading to those differences. iVariantGuide, you're doing a comparison, but in this case it's a single sample versus your reference sequence. And here we're looking at where the variants are and what pathways might be affected by those variants. So it's more of a analysis rather than a reverse analysis that we get with iPathway Guide.
Right. Let's see. Next question is, how do you evaluate the pathway/function impact of different variants affecting the same gene?
So each variant is evaluated independently. When we give you the pathway visualization, we show you the most impactful variant in that gene that is also in your filtered set of variants of interest. So if you filtered out some of the variants that co-occur in that gene, they will not show up on the pathway. So the most impactful variant that is still remaining in your variant set is what's displayed on the pathway.
Mm-hmm. Next question is, do we need to normalize the raw data before uploading?
So in the variant calling pipeline, usually there is some quality control that takes place, but it's not really a normalization.
Mm-hmm. Okay. Let's see, next question. Can we look at HERV-derived non-coding RNAs? I'm not familiar with that abbreviation.
There are some non-coding RNAs that are in our database, but I would say that that's pretty limited as of right now. So for the most part, we're looking at genes that are transcribed.
Gotcha. Question is, do you have API access?
That is another feature that is coming soon, and this will allow people like bioinformatics core facilities or CROs that are processing large volumes of data to automatically import and perhaps even do some early filtering using a command line interface. So yes, this is a feature that is coming very soon. We don't expect that it'll be something that everyone will want, but it will be something that some people want very much.
Yeah, all right, great. So we've kind of gone through the list of questions. As I mentioned, we will follow up with each and every one of you. I'll give you a few more minutes in case you have any other questions to ask while we're waiting for those last questions to come in. Just a reminder, if you happen to be attending the ASHG conference next week in Vancouver, please feel free to stop by our booth. We'll be in booth 414, which will be on the main aisle of the exhibit hall. So we would love to see you and talk with you and answer any other questions that you may have, even give you a personal demonstration and walk you through it. We'll have a couple of demo machines there that you'll be welcome to play around with. So I don't see any other questions coming in at this time. So I think with that, we're going to conclude today's webinar. We got done a little earlier than we had anticipated, but that's good. Enjoy the rest of your day and the rest of your week. And we look forward to seeing you at a future webinar event. Thank you.
Thanks a lot.